All right, so hi everybody. My name is Thomas Landrin, and I'm the co-founder of Lab Payas, which is a biohacker space that started a few years ago. And I'm going to give you a sort of story that is behind the concept and the phenomenon of biohacking. And for that, I'd like to give you a story, and that's actually the story behind this picture you see on the screen. The picture you see is not a simple childish scribble. It has much more meaning into it. And to explain you what it's coming from, I need to start 60 years ago, the story 60 years ago. What you see, it's those two guys are quite famous, they are Nobel Prizes. It's Watson and Crick. They discovered the structure of DNA. And by the discovery of this structure, we knew actually what the DNA, the matter of life, the information of life was made. So we could crack it. That means we could crack and hack it. That was really the beginning of modern biology. And in 60 years, biotech really has made jump and leaps, made huge advances. And biotech now is full of promises and revolutions. It's a very complex discipline. Biology is now becoming a technology, and we're in the way of mastering enormous amounts of information and mastering biological processes. But what we are right now, actually, in the story of biotech, make me think of the 20th century for the computers. In the 70s, sorry, in 20th centuries, in the 70s, at that time, computers were very big machines. They were only dedicated to a very specific task. And the market was basically 100 machines in the world. But at some point, because the know-how and the price of components were decreasing drastically, young students could gather together and think about computers in different ways. They could actually make their own computers. They could crack, they could hack, electronics and make their own computers for their own use. So by democratizing the access to computers, they created new kind of uses, and they basically bootstrapped the revolution we all know, under the name of internet and informatics in general. And if in the 20th century, people would crack and hack computers, in the 21st century, we hack biology. And this movement has a name. It's a global movement called do-it-yourself biology, or for DIY bio, or biohacking, or garage biology. And it's a very broad movement. It happens in labs, in open labs, what we call biohacker spaces. And here you have few examples. Basically, you have like Ireland, France, USA, even Syria. And you have biohacker spaces all around the world. It started in 2008 in the US. And really, really fast, it reached Europe. And as you can see, the two big dense points on the map are the US, basically North America and Europe. But it's starting actually to spread in Asia and in Australia. And there are some initiatives probably in Africa, and probably yet to come in South America. But why people are actually turning to biohacking? We have biotech, we have biology labs, and it's just, even now institutions doesn't have the ability to answer all our needs. When you're a young guy or a woman and you got crazy ideas, the institutions doesn't really have actually the mean to accompany you and give you actually the opportunity to find and fund your own ideas. This is a very simple example. It's basically the evolution of the average age for a person, for a researcher, to get its first grant in the US. Its first independent investigator grant. And if in the 80s you would wait, about at the age of 35, it's not bad, nowadays you have to wait until 50 or 55 even, which is actually, very shocking, especially knowing that Darwin and Einstein made their main discoveries before the age of 30. So this reappropriation of biotech, this global movement, actually is necessary today if we really want to invent new disruptive technologies using biology in general. And DIY bio can be defined by many things. But first of all, you have to define it using its values. And the first of its values is democratization. It's putting biology outside the wall of institutions. And it's opening the lab to everybody, saying that biology is too much important to leave it to the hands of professionals. Then you have interdisciplinarity. In biohacker spaces, the population you have is, because it's entirely open, you have as much scientists as non-scientists. You have, of course, biologists, engineers, informaticians, electronicians, but you also have designers, artists, jurists, lawyers, sociologists. And it's actually this whole diversity of people focusing on one single point, which is a new use of biology that makes actually change possible. Citizen counterpower. It's actually one of the main pillars of DIY bio is proposing biology as a counterpower tool. We live in a society where biotech is more and more, in the center of our life. It's not only medicine, but it's also the environment, the energy. And if we just want actually to live better with this technology, we better actually give the opportunity to our citizens to understand it and to be able to counteract, facts or give opinions. So if we want to live in peace with this technology, a counterpower is one of the major things to put into practice before getting further with the technology. Innovation, which is somehow associated with open source, because by opening, labs, by proposing actually biology to be in the hands of everybody, and by associating it with open source, which is basically the framework for people to share ideas and technologies, as you have in digital economy, for example, and digital innovation. Something you don't have in biotech. Just an example. You're 15 years old, you have a computer. Yes, you can write the best app ever. Electronics, somehow with Arduino and other platforms, more and more you can actually do such thing because you have open source access to modular technology that are well documented. But in biotech, it's not the case. You yet to have actually that kind of fundamental framework for you to actually decrease the cost of technology development. And that's what DIY Bio basically is doing right now. It's focusing on decreasing the price of technology, democratizing the technology itself by documenting, taking proprietary technology, cracking it, and giving it back to the community. Ethics and responsibility, finally, because when you're tackling with biology, it's not like playing with informatics or electronics. You're playing with life. So somehow it gives you a sense of responsibility when doing this. And that's why DIY Bio as a movement gathered a few years ago to create what even academics couldn't do, the first code of ethics for biotech. And as you can see, it's quite broad and give you very basic knowledge and sense of what is best and what is not for you to do. And in the very first position, you have transparency. Transparency is essential and it's connected to open source again. And again, because we know that we don't know everything, it's better actually for us to be entirely open and to leave others, look at what we're doing so that they can participate and give their opinions or even contribute. A few years ago, myself, I was starting my PhD in synthetic biology, and I heard of DIY Bio in the US. It was in 2009, and I thought, proning biology outside the world of institutions, being myself inside those walls actually made a lot of sense. actually seen, I've actually experienced a lot of frustrations and limitation of the system. So it made a lot of sense for me. So with friends, we founded La Paillasse, which is the first biohackerspace in France. And La Paillasse started in a squat. Why? Because for us it was really an experiment. We wanted to make a lab that was entirely free and open to everybody. We didn't have any money, and we surely didn't want to start it inside a university or a school or a research institute. It had to be entirely open. So we went to an artistic squat in Vitry-sur-Seine, and we found there a hackerspace called TMP one of the very first hackerspace in France. And we told them, we like very much what you do, but we'd like to do the same with biology. Could you give us a hand? And they basically opened us the whole community, and they explained actually the sense of hacking, which actually we didn't have because I was coming, for example, from biology and academics. I knew biology, but I didn't know hacking. So when speaking of biohacking, I couldn't even understand what was the meaning of it. So La Paillasse somehow is a biotech lab incubated inside the hackerspace. And this is what it became, because we actually made a lab entirely free, getting the whole lot of equipment you normally find in a lab for free. It's for more than 300,000 euros, only by getting free equipment from national laboratories. Because they have this sense of consumerism, that even themselves actually consume a lot of lab equipment, so that it's very easy to get your hands on it, even though it's still functional. And this is a few pictures of the lab inside the squat. And La Paillasse basically proposed for its community sessions for you to learn about theory and practices. You can experiment, we do workshops. For example, this is a workshop, what we call DNA barcoding. DNA barcoding is a very powerful methodology. It enables you to analyze the signatures, the genetic signature inside a sample. For example, it can tell you if you have GMO or not inside your food. Or it can tell you if your lasagna have horse meat or not. And the thing is that if you would like to do this yourself, you will have to send a sample of your food in a private lab. It will take about 3 days to have an answer and about 300 to 400 euros. So it's not really accessible. What we did is create what we call the quick and dirty DNA barcoding. It's a 3 euros per sample methodology for analyzing your own food. And again, just by figuring out how to crack the protocol and use the list of possible of components in your solution. We, of course, use, for example, DNA barcoding for other stuff like environment monitoring. We do experiment. Like, for example, we do our own lab hardware. Again, because we really want the lab hardware not only to get, to be used as a refurbished, example, but we really want actually for the lab equipment to be open source itself, so that it can be evolving inside the community. We organize debates about various questions. And showing you all this about La Paillasse actually somehow is already the past for us, as the lab in a squat was a prototype. We wanted to show that such a framework for creativity, counterpower, innovation was actually something real, that it could be done. And what we did is that from our squat, we went to see the mayor of Paris and said, should probably take La Paillasse as its principle and put it right in the center of Paris, because if you do that, this is going to become a real bomb, like a catalyzer for innovation, because then everybody could access biohacking, and not only students, but all the professional network of the Parisian region. There are several hundreds thousand students in Paris. And that's actually what we did. So I'm very happy to say that since a month now, La Paillasse just got a huge new space in the center of Paris, right in the Sentier. And so basically, we came from that to that. So it's a big change for us. And there's a few pictures. Now it's not empty anymore. This is the first floor, and this is underground. So yeah, somehow comparing this to the squats, it seems like a bit too much proper. But we got a beautiful cave for us to get our lab installed. So we won't be lost totally. And so the plan actually is to build the largest biohacker space we know in the world right now. It's 750 square meters, and basically you'll have everything from the space for you to work to the labs with biotech and fab labs. So you'll be able to address questions around software, hardware, and wetware. And right now, La Paillasse is because it's restarted small and as a proof of principle, and that finally we could find a working solution for biohacking in Europe, it started actually to replicate. And quite fast, actually, we are now getting Paillasses in Bordeaux and Lyon, and one also in Lausanne, one in Manille in Philippines, and one in Cork in Ireland. And at the end, what we really want to do is create a sense of network with an organized community that can work together wherever they are, and they can connect to each other so that they can work and crack all the biotech they can find and figure out maybe what can be the innovation of tomorrow. But how do you actually equip so much labs? Again, those are very low-cost labs. We organized what we call la campagne des biochineurs. Basically, it's like biotech antiquing. It's going finding old lab pieces of equipment. And there are thousands of labs around France. But how do you actually access to all of them? The logistic will be enormous. So we basically actually use our community so that we can address all those issues. And it's easy for them to just go to a lab, transport a lab equipment, and bring it to a Paillasse. And it makes it the thing entirely free, very agile, and enables the creation of many Paillasses around the world. So now I'd like to give you a few examples of what we are doing. So here, for example, is a very fundamental machine you find in biotech lab. It's called a PCR machine. PCR machines are what makes you give you the ability to analyze DNA. Normally it costs around at least 5,000 to 10,000 euros. But this project called the Open PCR project bring the price down to 500 euros. So this is actually the kind of technology, by democratizing the access to lab equipment, that will actually change the way biotech evolution and biotech innovations can be done. This is just the start of something much bigger. And to give you other examples very fast, this is another project called Amplino. It's a diagnosis machine for malaria. It's made in a very DIY framework. As you can see, it's made in a shoebox. And for only 50 euros, the machine was made, and it enables somebody to analyze a sample of blood for the presence of malaria or not for only $1. Now they have a new prototype. They got funded, and they are going to actually test the machine on the field in Africa this summer. This is another example. Two engineers from Sony, they went to work at La Paillasse. They wanted to work with biodegradable electronics, and they didn't have any knowledge of biology. They knew electronics and informatics. So basically help them, to understand all the process. And we work with them to find a good solution for them. And actually we found a way to produce a biomolecule that has semiconductive properties. And to work with semiconductors, you need to work in a very neutral atmosphere and without oxygen, basically. But for that, you need a glove box. The glove box are usually very expensive, and we couldn't actually get the hand on one that was refurbished. So those guys basically made a glove box from an IKEA box, and it works perfectly fine. We work with designers and artists. For example, this guy wanted to work with biological patterns, and how actually we could master the life of a living organism so that it can draw something out of it. And he used a bacteria that can, migrate toward the source of nutrients that you put on a petri dish. And by putting those sources of nutrients very strategically, he could actually create a pattern in the form of letters. And he made all the letters of alphabets, making at the end a biological graphical font. There are now people working with what we call biomaterials. Biomaterials are very interesting because basically you can produce it yourself. You only need basic components. You provide yeast or bacteria, and they process those basic components, transforming into biomaterials. Like this, for example, this is celluloses. Celluloses can be used, for example, for making clothes, like this one. And this brings me to the last part of my story, which is actually the beginning, is how this drawing was made. And it has a link with this little piece of earth, right? So would you have any idea what's the link between them? The link between them is a petri dish of bacteria. Those bacteria are found in the soil, and they produce a blue pigment. The idea behind this project was, is it possible to make a pen that produces its own ink? So instead of having a cartridge, you have a bioreactor inside with bacteria that is producing the pigment that you use for writings. In exchange, you feed the bacteria. And at the end, it's like feeding your own pen. We worked on this project with a designer, and we came to the point where we actually understood that it was possible. And we found a process to use the natural bacteria to produce out of it a biological ink. It's biodegradable, it's non-toxic, it's a blue ink. It can be used in any pen, and you can produce it yourself very easily. It can even change colors. It can turn to red, to green, to yellow. And with the designers, we actually work on a kit so that it would be easy for anybody to grow and make its own ink at home. And this is just an example we had with a little workshop we organized. And so this is one of the very first versions of the kit we designed with our team. And we really wanted to bring the context of biology outside the lab. That's really important for us. And but this whole reflection about biotech and biomaterials brings me to a very serious point I'd like to finish on. It's, we live on a planet which is like a closed system. It's like a spaceship. You have a limited amount of materials and energy. So you have to find processes so that you limit actually this waste. So that's how actually we came to the idea that maybe biomaterials are the solution. They are easily produced and they can easily degraded. So why not actually pushing forward that kind of solutions, not only for your own purpose, but for much drastic purposes. Like for example, that's how we got into contact with NASA, and we decided, how do you actually produce massively and modularly biomaterials? It's not only using petri dishes like that. You have to use bioreactors. Bioreactors are very expensive machines. They are costing at least 15 to 20,000 euros. So we told NASA basically, you want to spend humans and ships in space, but how are you going to get that far if you actually don't have a way to recycle everything you have on the ship? So that's how actually NASA decided to give us some money so that we can work on a cheap open source bioreactor that could be open source, and so that we could build a community around it and basically democratize this technology in order to create a new kind of uses. And for me, bioreactors are somehow the third link of a big chain. We all know today that we have access to data. You know, we are now getting into the era of 3D objects, and 3D objects are just the step before getting into manufacturing and the ability to manufacture anything you want as biomaterials that can be used afterwards for making objects. So, being a biohacker, for me, is being a pioneer for humanity. You're here to test everything, to test new stuff, to actually drive a new way of innovation in biotech, a way that institutions right now are not able actually to provide. And anybody can be a biohacker. And the first thing you need to do in order to become one is to reclaim what's already yours. It's life. Thank you.